Today we'll be talking about DNA replication. Now, as we know already, DNA consists of two complementary strands of nucleotides. The DNA splits, and each of those strands is going to serve as a template to make the missing strand. Of course, things are more complicated than that. Life is messy, things are overly complicated, and this has to be done in several steps, as we'll see. Once we talk about how DNA is replicated, we can talk about how viruses work, which I think is quite fascinating, and that will be our next topic. If you think back to when we started this whole journey on DNA, I mentioned Watson and Crick briefly. Watson and Crick were the researchers that figured out the structure of DNA. They didn't do that much in the way of hands-on research. Instead, they looked at the research by a lot of other people and they consolidated it. They brought it all together to make a working model of DNA. And right off the bat, in their short little paper, what they said was, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So what were they talking about? They were talking about the fact that we have these two strands of nucleotides that run parallel to each other, and the nucleotides are complementary, so the bases are complementary. If we have an adenine on one side, we have a thymine on the other side. If we have a C, which is a cytosine on one side, we have a G, a guanine on the other side, etc. So we have these two strands that are held together by hydrogen bonding. Hydrogen bonds are fairly weak, but there's a lot of them. So the molecule as a whole is fairly strong, but each one of those pairs of nucleotides can be pulled apart quite easily. And this is exactly what happens. So the two strands are separated just a few nucleotides at a time. Once the strands have been pulled apart, an enzyme known as DNA polymerase comes along and it builds the complementary strand for each of our parental strands. So we end up now with two separate double-stranded DNA molecules, and each one of those molecules consists of a parental strand, shown in dark blue here, and a daughter strand, shown in light blue. In addition to the strands being complementary, they are also anti-parallel. Have a look at the zoomed in portion at the top there, where we're looking at basically one rung of the ladder that is DNA. We have a phosphate, we have a sugar, we have an adenine on the left-hand side, and on the other side we have a thymine, a sugar, and a phosphate. But notice that the thymine and the sugar and the phosphate are upside down compared to what we see on the left-hand side. This is the anti-parallel part. The nucleotides that make up DNA, of course, have three parts. We have our nitrogenous base, so that would be the ATCG, and then we have our sugar, which is deoxyribose, and then we have a phosphate. And then these nucleotides are brought together to make a single strand of the double-stranded DNA molecule. Let's take a look at the sugar. So the sugar is shown at the top of this diagram here. And we have 5 carbons. The sugar is a cyclical structure. It's a ring, basically. So we have 4 carbons that are found in that ring, along with an oxygen, and then we have 1 carbon that lies outside of that ring. And the carbons are numbered 1' through 5'. So that little single quote mark, that means prime. We start with our first carbon here, our 1' carbon, and it is attached to the nitrogenous base. You can see that down below here. So we can see in this case, it's attached to an adenine. Then we have that carbon attaching to our second carbon, which is attached to our third carbon, which is attached to our fourth carbon, which is attached to the 5th carbon, which lies outside the ring, and that 5th carbon is attached to a phosphate. DNA strands are built in a very specific way. So what happens is we bring in new nucleotides and we add that phosphate that's sticking out from the incoming nucleotide to the 3' hydroxyl. So that's the OH group that's attached to that 3' carbon. And that's the only way we can build a strand of DNA. So it's not shown here, but there would be a hydroxyl here, and that's where we're going to add the next strand onto. Because of this, we have to build the two strands in opposite directions, and that's because they're anti-parallel. Again, that's because the orientation of one strand is upside down compared to the orientation of the other strand. We'll have a closer look at what happens when we build a single strand of DNA. We've got two deoxyribose sugars here, and you can see they're linked together through a phosphate. At the bottom of this chain, we've got a free hydroxyl group. A hydroxyl is an OH, an oxygen-hydrogen group. That's the only place we can add a new nucleotide. The nucleotide coming in has 3 phosphates attached to it. 2 of those phosphates are going to be pulled off, and we're going to create this new bond. Again, the really important point here is that we can only build onto that free 3' hydroxyl. The site where a DNA molecule is split apart and replicated is known as a replication fork. So we have our original DNA molecule at the top there with 2 parental strands, again shown in dark blue. We have the area or region of replication where things are being pulled apart and complementary nucleotides are being added. And then down the bottom we have our newly formed DNA molecules that consist of a parental strand, again dark blue, and a new strand, a daughter strand, which is light blue. We'll talk about how that whole anti-parallel thing complicates everything in just a moment, but before we do, let's look at the overall process. And we'll start with the simplest case, which is what happens in bacteria. The DNA in bacteria is always in a circle. We have one giant circular chromosome that contains most of the genes, and then there's a handful of other genes that are found on tiny circles known as plasmids. Whether we're talking about a plasmid or the chromosome, we always have an origin of replication. And that's where the DNA is pulled apart. That's where replication begins. So there are enzymes that we'll talk about that will pull the 2 parent strands, and they're shown in dark blue here, apart to allow the creation of the daughter strands, shown in light blue. Pulling the strands apart at the origin of replication creates what's called a replication bubble. And then replication is going to proceed in both directions away from the origin of replication. This creates a replication fork at each end of the replication bubble. The replication forks proceed in opposite directions around the loop of DNA, and eventually they meet up. And then the circle of DNA will split off into 2 new daughter DNA molecules. Each of those daughter DNA molecules consists of 1 parental strand and 1 daughter strand. You can see this actually happening in the image at the right there. This is an electron micrograph taken by a scanning electron microscope. So basically it gives us a 3-dimensional image of what the DNA actually looks like. What we're seeing here is a plasmid that is dividing into 2. Here's an example with E. coli. E. coli is a very common bacteria. You've probably heard of it. You have lots of it in your digestive tract. It has one very, very large chromosome that contains about 1,000 genes or so, so the majority of the information that's found in the cell. And it has a single origin of replication on that single chromosome. In addition to the chromosome, it may have several plasmids. The plasmids don't have nearly as many genes on them. This is not to scale. Plasmids are much, much smaller. But the plasmids generally have 1, 2, 3 or so genes on them. The plasmids also have a single origin of replication. Now, what's really quite fascinating is that we can trick bacterial cells like E. coli into copying foreign DNA. If we take a piece of DNA, and it could be from anything, it could be from a plant, it could be from a human, if we take that piece of DNA and we bend it into a circle, so we take the ends of a straight piece of DNA and stick them together to make a circle, and we also insert an origin of replication, so it's shown here as ORI, we take that new molecule and we stick that into E. coli, it will blindly make copies of it. Because the enzymes found in E. coli are just looking for that origin of replication. When they see it, they split the strands of DNA and they copy them. And every time the cell replicates, it will replicate this DNA. As we'll see, this is pretty important when it comes to genetic engineering and so on. We can use bacteria to make copies of DNA, and we can even trick the bacteria into decoding that DNA and spitting out the appropriate proteins. Here we have an actual image taken by a scanning electron microscope. When you take a picture, quote unquote, with an electron microscope, you get a black and white image, but they've added some colors here to make it a little prettier. But we're seeing loops of DNA. Now, the DNA doesn't usually exist in just a simple loop. Usually it's supercoiled. And what that means is it's further twisted. So imagine you take a rubber band, which is a simple circle, and then you take that and you twist it and twist it and twist it. That's usually how circles of DNA exist in bacteria. They just take up less space this way, and also they don't break as easily. That supercoiling doesn't occur in eukaryotes. We have histones. Remember, we have these positively charged proteins that can stick to the DNA and help condense it and protect it, but prokaryotes don't have that. The big difference between prokaryotes and eukaryotes, though, when it comes to DNA, is that the DNA in eukaryotes is not in a circle. It's straight. So you have 46 chromosomes, and each one of those chromosomes consists of a single piece of DNA that's wrapped around these histones and condensed. You do not have a single origin of replication for each of your chromosomes. Instead, there's multiple origins of replication, and that's because we have a lot more DNA. If we just had one origin of replication in the middle, let's say, of a strand of DNA, and the replication forks proceeded to the outside, it would just take too long. So we have several origins of replication on each chromosome. Just like we saw in prokaryotes, at each of the origins of replication, we have a bubble being produced as the parental strands of DNA are pulled apart. Each bubble has two replication forks again, and those replication forks move away from the origin of replication. Eventually, those replication forks will meet up with each other, and the bubbles will fuse, and we'll get two complete daughter DNA molecules. Once again, we're going to have a parental strand for each of those DNA molecules and a daughter strand. What you're seeing on the right here is what this looks like under the electron microscope. In eukaryotes, we can see that we have a bubble here, we have a bubble here, and we have a large bubble over here. The arrows are depicting replication forks. There's also replication forks here that are moving out of this bubble, and all of these bubbles will eventually fuse together. Another image of the same thing. The arrows here are pointing out 5 replication bubbles on this electron micrograph. Okay, so let's take a look at one of these replication bubbles in a bit more detail. We have the origin of replication, and that's where the bubble first opens up. And then we have two replication forks moving away from that origin of replication in opposite directions. Remember that every strand of DNA has a polarity to it. It has a 3' end and a 5' end. The 3' end is where we have that hydroxyl group. So we can build off of that end. We can't build off of the 5' end. Now what happens is at each fork we have what's called a leading strand, and it can be built in one continuous piece, and then we have a lagging strand because it lags behind, it takes longer to replicate, and it's built in short little pieces. The red primers here are short little sequences of RNA that are put down first that the DNA can be built off of. So the enzyme that builds DNA, DNA polymerase, can only build off of something that's pre-existing. It can't simply build off of nothing. It needs to have a short little strand of something, RNA nucleotides in this case, that it will build off of. It needs a 3' end to build off of. And that'll make a lot more sense after we go through each one of the steps. I'm going to start by doing a quick overview of the entire process. And please don't judge me or laugh at my drawings here. I'm trying to do this freehand. But we have our two parental strands of a double-stranded DNA molecule. And I'm going to draw them here. This is our replication bubble. So this is where the DNA is opening up. Again, my drawing skills are not great. For the sake of argument, let's say that this particular DNA molecule has a 3' end here and a 5' end up here. So remember that each strand has a polarity to it. We can build off of a 3' end, we can't build off of a 5' end. The strands are anti-parallel. So that means this strand here is going to be 3' and it's going to be 5' over here. It's running in the opposite direction. So we have our replication bubble. Our origin of replication would have been around here in the middle. That's where things originally opened up for us. And then we're going to build off of that. When we build our daughter strands, we use a few different enzymes. Again, it's more complicated than you might think. The first enzyme is an enzyme known as primase, and it makes something called a primer. And this is something that we can build off of. The primer is not made of DNA, it's actually made of RNA. So we have a short little primer here that's made of RNA, and that RNA is complementary to the DNA. So the DNA strand at the top is complementary to that short little strand of RNA that I've just drawn. If we look at the polarity of that, again, the same rules apply. So this is going to be the 3' end here. This is going to be the 5' end over here. We cannot build off of the 5' end. The 3' end has a hydroxyl sticking out that we can build off of. The next enzyme is DNA polymerase. Specifically, it's a form of DNA polymerase known as DNA polymerase 3. It can't build off of nothing. It needs to build off of something. And fortunately, we have that RNA primer with a 3' end sticking out. And it can build off of that 3' end. It can't build off of the 5' end. So what's going to happen is DNA polymerase is going to build off of that 3' end in this direction. And once it starts going, it's going to keep going. So as the replication fork opens up to the right here, it's going to just keep going. It's going to add complementary DNA nucleotides. It's going to read the sequence on the top, on the parental strand, and add the complementary DNA nucleotides. And it's going to just keep going. That's known as the leading strand. Now let's take a look at the other strand of DNA. It also has to be copied, of course. Same thing is going to occur. We're going to get a short little piece of complementary RNA being laid down. And just like before, we have to follow the same rules. So this is going to be our 5' here. This is going to be our 3', and we can only build off of the 3'. So we're going to build off of the 3' towards the left. So in this direction, and we can keep going as that replication fork opens to the left. This again is the leading strand. Now we have a bit of an issue going in the other direction on each of these strands because we can't build off of that 5'. So what happens is RNA polymerase comes along and it skips ahead a bit and it lays down a primer here and it lays down a primer here. And again, following the same rules we've talked about, we'll add in the direction here, 5', 3', and I'm sorry that it looks like a child wrote this, but you know, doing the best I can here. 3' and 5'. And now DNA polymerase 3 can come along and build off of that. So it can build these short little pieces from here to here, and from here to here. There's a little gap left there. That RNA has to be broken down and removed and replaced with DNA. There's another enzyme that does that. It's another DNA polymerase. It's DNA polymerase 1. And then there's another enzyme that has to come along and glue everything together. And that's something known as ligase. But what we have is we have leading strands that are kind of one-shot things. So they'll just keep going. And then we have what's called the lagging strand. That's the one that's built a little bit at a time. And it's more complicated. It takes a bit longer. It kind of holds up the whole process. Hence the name lagging strand. And I do have to admit, this drawing's a little better than mine. So looking at this again, for any particular replication fork, we're going to have a leading strand and a lagging strand. So that's one replication fork. We have, in this case, the lagging strand on the top, the leading strand on the bottom. On the opposite side, the replication fork on the left, we've got the same thing, but now the lagging strand is on the bottom and the leading strand is on the top. We'll take a look at each of these strands in turn, and we'll start with the leading strand because it is the simplest one. So this is just one of those odd quirks of evolution where enzymes are very specific in what they can do. DNA polymerase builds DNA, but it can't build off of nothing. It can't read a strand of DNA and just start laying down complementary nucleotides unless it has a 3' that it can add to. Fortunately, there is an enzyme known as primase that can build off of nothing. It can build off of this DNA template without having a pre-existing nucleotide to build from. Primase is going to lay down RNA nucleotides, and that's what the red is here. So it's going to read the DNA sequence, and it's going to lay down the complementary RNA nucleotide. So if it sees a cytosine, a C, it's going to put a G, a guanine. If it sees an A, it's not going to put a T because remember we don't have T's or thymines in DNA, we have uracil instead, we have U. So if it sees an A, it's going to put a U, etc. It's going to build this complementary strand of RNA along that DNA template. Now, the next thing that has to happen is DNA polymerase is going to come along, and now it has that 3' end. It doesn't matter that it's RNA. It has something to build off of. It's going to build off of that 3' end using the DNA, single-stranded DNA template, to add the appropriate DNA nucleotides. Now, if we want to be absolutely correct, this particular DNA polymerase is known as DNA polymerase 3, and that's supposed to be the Roman numeral 3 there. There are lots of different DNA polymerases that do slightly different things. Some of them build DNA, some of them repair DNA, etc. Unfortunately, the DNA polymerases were named in the order that they were discovered and not necessarily the order that they work in. But DNA polymerase 3 is going to build off of that RNA primer. This builds the leading strand. It can just keep going because it has this continuous 3' end to build off of. The next thing that needs to happen is that RNA primer has to be replaced with DNA. So the whole goal of the cell here is to build a DNA molecule. We don't want DNA and bits of RNA. We want just pure DNA. So now we have an enzyme. And again, if we want to be technical about this, it's DNA polymerase 1 that's going to come along, and it's going to replace the RNA primer with DNA. It'll go one by one nucleotide by nucleotide. It'll replace the RNA nucleotide with the appropriate DNA nucleotide until that RNA primer is completely replaced with DNA. So this is what that looks like. There's a few other enzymes here that we'll come back to and talk about again, but we have an enzyme known as helicase, and it actually unzips the DNA. It separates it into 2 strands. We have single-stranded binding proteins, which are just proteins that stick to the DNA and stop it from coming back together. We don't want those 2 parental strands just coming back together and forming new hydrogen bonds. We want to keep them separate. We have an enzyme known as topoisomerase, which helps relieve any twisting in the DNA. And again, we'll come back to that in a moment. But anyway, have a look at primase there. Primase is adding this RNA primer. It's adding this short little primer that DNA polymerase can build off of. Next, DNA polymerase is going to come along. It's going to clamp to that parental strand of DNA, and it's going to build off of the 3' end of the RNA primer. That is the leading strand. We can see that on the top here. It's the top strand in this diagram. And it's going to just keep going. So helicase, that green enzyme, is going to keep unzipping the DNA. So the replication fork will keep moving to the left. And as it does that, DNA polymerase 3 just keeps building and keeps elongating that new daughter strand. Now let's take a look at the other strand, the lagging strand. This one is a bit more complicated because it has to be built in short pieces as the replication fork opens up. For each replication bubble, once again, we have 2 replication forks. One strand is going to be the leading strand, one strand is going to be the lagging strand. So just like before, primase comes along and it adds a short little RNA primer. That gives us a 3' end that RNA polymerase 3 can build off of. It's going to build a short segment of DNA, and that's known as an Okazaki fragment, and that's named after the Japanese researcher that first discovered this. DNA polymerase 3 is then going to detach when it gets to the start of the next fragment. It's going to bump in to a previously laid down RNA primer. As the replication fork continues to open up towards the left in this example, we're going to have a new primer laid down. So we've got a new RNA primer being laid down, and then we've got this new Akasaki fragment being built off of that. Eventually, it will bump in to the old primer. And when it gets to the old primer again, that DNA polymerase 3 is going to detach. It's going to leave. Now we're left with two issues that need to be resolved. The first thing is that primer needs to be replaced with DNA, because that primer, shown in red, is made out of RNA. Just like before, DNA polymerase 1 will come along and do that. That's not really a big issue. But the other issue is that we need to seal the gap that results. So after that RNA has been replaced by DNA, we still have a gap here. DNA polymerases have a couple of shortcomings. So DNA polymerase 1 can replace RNA with DNA nucleotides. That RNA has to be there first. And DNA polymerase 3 can lay down DNA nucleotides directly using another strand as a template, but it needs to build off of a 3'. It needs to build off of the primer. There isn't a DNA polymerase that can seal that little nick. There isn't a DNA polymerase that can attach a 3' end of DNA to a 5' end. This would be a 3' end here, and this would be a 5' end here. And there is no DNA polymerase that can fix that. But fortunately, there's an enzyme known as ligase. And ligase will basically glue DNA together, and it will seal that nick. And that's what this kind of turquoise enzyme you're seeing here is doing. That's ligase, and it's going to fix that little gap. Once again, for any replication bubble, we're going to have two replication forks moving in opposite directions. Each replication fork is going to have a leading strand. So the diagram on the bottom there, the leading strand is on the top. It just needs to be primed once, and it's going to be a straight shot in one direction. It's going to just keep going in the same direction as the replication fork opens up. On the other strand, we have our lagging strand. And this has to be built in short pieces. It has to be primed multiple times. We build off of the primer, we build these Akasaki fragments, and then the fragments have to be glued together using ligase. We've talked about a few very important enzymes when it comes to DNA replication. We have primase that adds a short RNA primer, and that primer allows DNA polymerase to have something to build off of. We have DNA polymerase 3, which builds off of the primer, and DNA polymerase 1 that replaces that RNA with DNA. We also have ligase, and ligase will seal up the gaps. It basically glues the Akasaki fragments of the lagging strand together. There are some other important enzymes and molecules, though. Helicase is responsible for breaking the hydrogen bonds between complementary DNA strands at the origin of replication. So this is what creates the replication bubble and what allows the replication forks to move away and expand that bubble. Helicase is also known as unwindase, which I think is the coolest and also the most ridiculous name for an enzyme ever. But it unwinds the DNA, separates it into separate strands that can serve as templates. After it's done that, we have these single-stranded binding proteins that bind to the single-stranded DNA to stop it from zipping back up right away. DNA strands, if they're complementary, will come back together naturally. That's a more stable situation. But if we have these single-stranded binding proteins, the DNA will form bonds with it instead of with the adjacent strand. Helicase basically expands the replication forks. Basically, it just pulls the DNA strands apart. Now imagine if you had two strings or two ropes wrapped around each other like a molecule of DNA, and you just pulled on those two strings. What would happen is everything would get tangled up ahead of that fork. There's another enzyme known as topoisomerase, which goes ahead of the replication fork as it moves along, and it actually cuts the DNA. It cuts one strand of the DNA, moves it around to relieve this excess coiling, to relieve any loops and tension and so on that might result as an effect of pulling those two strands apart. Let's have a look at how these enzymes work together by looking at a short animation. The very first step is that the enzyme helicase, or unwindase, comes along and looks for the origin of replication. So there's a sequence of nucleotides that it looks for specifically. It will unzip the nucleotides, and then single-stranded binding proteins come along to ensure that those two strands stay separated. The ultimate goal is to make new daughter strands of DNA. DNA polymerase III is responsible for that. It adds nucleotides to the 3' end. Now, once again, it can't do this if there's nothing to build off of. So what happens first is primase comes along and it adds an RNA primer. This gives us a 3' end that DNA polymerase III can build off of. And it's going to read the parental strand of DNA and build in the 3' direction. That is the leading strand. Now we have another strand that we have to deal with as well, but remember that DNA is anti-parallel. So we have to build the other strand in short little chunks, and that would be the lagging strand. So as the replication fork opens, so we have helicase there opening it up, we have primase coming in, adding a new primer, DNA polymerase III building off of that until it hits the old primer. Then we have DNA polymerase I coming in and replacing that RNA with DNA, and then DNA ligase coming in and sealing that nick. So connecting those Okazaki fragments together. And this happens over and over again. So the speed of this process is held up by the lagging strand. It can only proceed as fast as the lagging strand can get things done, hence the name lagging strand. This is what it really looks like. Again, we don't want to get into too much complexity here, but we have enzymes that are actually all clumped together. They're clumped together in a big complex. So we have our leading strand at the top there, our lagging strand at the bottom, and keeping all of these enzymes linked together like this keeps things going at a set rate. It makes sure that the leading strand and the lagging strand can proceed at the same rate. There's one last problem to deal with. What happens when we get to the end of the piece of DNA that's being replicated? For prokaryotes, like bacteria, there really isn't an end because their DNA is in a circle. So the two replication forks will eventually meet, and when they do, DNA ligase will be used to glue the new strands together. But that's not what happens in eukaryotes because we have straight pieces of DNA, or linear pieces of DNA. And this actually results in something interesting. Our DNA erodes over time. What I mean by that is we lose a little bit from the ends of our chromosomes every time our cells replicate. This does not occur in bacteria. At the end of a straight piece of DNA, An interesting problem arises when the cell tries to make a copy of that DNA. And we can see that here. So we're zoomed in at the end of a piece of linear DNA. The leading strand does not present a problem. For the leading strand, it will just continue right to the very end. But with the lagging strand, of course, we have to build that in small fragments, and it requires that an RNA primer be put at the end of the DNA molecule. That RNA primer is going to have to be replaced with DNA. That occurs with the help of DNA polymerase I. Let's take a look at the second RNA primer in the second part of this diagram. There's not an issue here when DNA polymerase has to replace that RNA primer because it has a 3' end to build off of. So it's going to build off of that 3' end. It's going to go nucleotide by nucleotide and replace the RNA with DNA. Now when it gets to the end of that RNA primer, there's going to be a gap left. But again, that's okay because ligase will come along and it will seal that. And that's what we see has happened in the next diagram. However, that very first RNA primer, the one at the very end, presents a big issue. We don't have anything here. We don't have a 3' end that the DNA polymerase I can build off of. So that can't happen. We cannot replace that RNA primer with DNA. Instead, the RNA gets broken down. RNA is kind of delicate. There are these enzymes known as RNases that wander around and gobble up RNA. The RNA is broken down and it's lost. We have a gap at the end of that piece of DNA. When the cell divides again and the DNA is copied again, we don't have anything to copy at the top. So the DNA gets shorter. Through each division of the cell, the DNA erodes. It gets shorter and shorter and shorter. Now, if we had important information on the ends of the DNA molecule, we would be losing that information, and that would be rather bad. The way our cells deal with this loss of DNA is that they add a bunch of junk to the ends of the chromosome. So the ends of the chromosomes contain nonsense. There is no information there. There's an enzyme known as telomerase, and it builds telomeres. And these are the junk bits of DNA at the ends of the chromosomes. This junk DNA contains the sequence TTAGGG over and over and over again. And there's a reason for that. That's because telomerase carries its own template with it. It's got this built-in chunk of RNA, and the telomerase enzyme will travel along, and as it does, it adds this repeated sequence, the complementary sequence to that RNA, to the end of the chromosome. And this means that the cell can divide many times, and the DNA can erode, and it will take a very long time before it erodes through all that junk and gets to actual important information. Here we're seeing a set of replicated chromosomes. So each chromosome contains 2 chromatids in this case. And what they've done is they've taken a piece of single-stranded DNA that will recognize that specific sequence that we see in telomeres. So it has the complementary sequence, it binds to it, and this piece of DNA that they've created, it's called a probe, it has a fluorescent dye attached to it. So we can see where the telomeres are located here at the ends of the chromosomes. Interestingly, telomerase is not active in all of your cells. It's only active during early development, and it's only active in the cells that would be set aside in the gonads, in the testes and the ovaries, to produce gametes. When your father's sperm and your mother's egg came together, they had these telomere sequences at the end of every chromosome. So the chromosomes were lengthened with this non-coding junk. As a zygote, you had these long telomeres, which allowed for your chromosomes to shrink every time your cells divided during development. Essentially, you have an expiration date stamped on you when you're a zygote. Your cells can only divide about 200 times before those telomere sequences are gone and you start to lose important information. You start to lose genes. And it's thought that this is what causes aging, at least one of the things. So your cells have the ability to repair themselves and maintain themselves, but once they start to lose information, they can't do that anymore. Prokaryotes, like bacteria, because they have circles of DNA, they don't have to worry about that. And in fact, those cells are immortal. So bacteria cells do not die of old age. They die for other reasons, but not because of old age. This is kind of a fountain of youth in some ways. So it's thought that maybe if we could find a way for body cells to also use telomerase, perhaps we could live a lot longer. Now, there might be some problems with that. The longer you stick around, the higher your chances are of developing mutations in your DNA. And eventually, you will develop mutations that can't be repaired. Telomerase can also become active in cancer cells. And there are some cancer cells that are immortal because they keep lengthening their chromosomes. We have cultures of cells, human cells, that are used in research that I'll talk about later that are immortal. We can keep dividing those cells over and over and over again, and they keep lengthening their chromosomes. In cloned animals, which is what you're seeing here, they tend not to live quite as long as non-cloned animals. And that also appears to be a result of the fact that we're making these animals from cells that have already shrunk their telomeres quite a bit. We're cloning animals from adult cells, which is an issue. But these are issues we'll talk about when we talk more about genetic engineering and so on. This diagram is a nice overview of the whole DNA replication process. We've got helicase, which is creating a replication fork. It's unzipping the DNA into two strands, and the replication fork in this diagram is moving towards the left. Immediately after the strands are detached from each other, we have single-strand binding proteins that bind to the strands and stop them from immediately reforming those hydrogen bonds. Upstream of this, we've got topoisomerase, also known as gyrase, that will cut one of the strands, rotate it around, seal it back together again, just to relieve any twisting that's caused by helicase. And then, of course, we have our leading strand and our lagging strand. The leading strand is a one-shot deal. The lagging strand has to be continuously reprimed, and it's built in segments. Now, I was going to talk about some of the applications of this today, the polymerase chain reaction. I was going to talk about how we could use ligase to stick pieces of DNA together and build interesting arrangements of genes, but I think I'll come back to that a little bit later. In summary, here's a few of the important concepts from today. A DNA molecule consists of two strands, and the strands are complementary and anti-parallel. We can only build off of the 3' end of a strand. That's the only place we can add new nucleotides. And that results in there being a leading strand and also a lagging strand that's built in short segments called Okazaki fragments. There's a whole bunch of enzymes involved in this, and we talked about DNA polymerase I and DNA polymerase II. We talked about single-strand binding proteins that keep the strands separated from each other. We talked about ligase that seals nicks, and we talked about helicase that unzips the DNA to create a replication bubble with 2 replication forks. There's a number of other enzymes that are involved in repairing DNA as well that we didn't get into. All of these enzymes are quite important when it comes to manipulating DNA artificially as well. And we'll talk about that later. We'll talk about how we can make artificial pieces of DNA, how we can take DNA from 2 organisms and stick them together with ligase. Ligase can be used as glue to do that. We'll talk about how we can make artificial plasmids and so on. Telomeres are added by an enzyme known as telomerase to the end of linear DNA, just in the cells that give rise to gametes. And that's to prevent loss of information caused by chromosome shrinkage or erosion every time a cell divides. And finally, here is our terminology for the day. We're going to come back and revisit some of this stuff again, look at its applications in biotechnology, and also some of this will apply to how viruses work, which I think is quite a fascinating topic. See you then.